Hello friends, I am Dr. Ashish Agrawat. Welcome to my channel Live Biochemistry. In this video, I am going to discuss about the lipoprotein metabolism. There are two types of cycle in lipoprotein metabolism. One is exogenous cycle and one is endogenous cycle. Exogenous cycle is mainly taken care by the chylomicrons, that is the lipoprotein, right? And it will carry the dietary lipids from intestine to the peripheral tissue. While endogenous cycle is mainly by the VLDL, HDL and LDL, right? So you can see here whatever diet we take, dietary lipids that we take, either in the form of oil or butter, when they are absorbed, these lipids are absorbed along with the chylomicrons and they will be transported to the peripheral tissue. While VLDL, HDL and LDL, they will transport the endogenously synthesized lipids. So now we are going to discuss about the chylomicron metabolism. So if we see the structure of chylomicron here, because lipids are insoluble in water, they are hydrophobic, so they require the protein. For the exogenously supplied lipids in the form of triglyceride, phospholipid or cholesterol, they will combine with the ApoB48. This ApoB48, this is the apoprotein which is synthesized by the intestinal cell, by RNA editing. So here in the liver cell, there is ApoB100 is synthesized. The gene for the synthesis of ApoB48 is same as ApoB100. But what happens in the intestine? There is a deaminase enzyme. So this ApoB100, it is made up of 4, 5, 6, 3 amino acids. Right? In the intestine, up to 2152 amino acids are synthesized, and when 2153 amino acid, okay, the codon for this 2153 is CAA. But in the intestinal deaminase enzyme, it will convert CAA to UAA, that will be stop codon. So in the intestine, up to 2152 amino acids are formed, and it is 48% of 4563. That's why this apoprotein is called as the ApoB48. Okay, so if someone asks which is a major apoprotein in chylomicrons, that your answer should be ApoB48. That is frequently asked in competitive exams. And the highest concentration of triglyceride is present in the chylomicron. So highest triglyceride in case of chylomicron, that you have to remember specifically. Right. Now coming to the metabolism. So you have already learned in digestion and absorption of lipids. Suppose this is a intestine. Right. So intestine lumen contains the triglyceride, phospholipid and cholesterol ester. They are broken down by the pancreatic lipase to glycerol, fatty acid, cholesterol ester, okay, and they will aggregate to form the mission. This mission will be absorbed in the intestinal cell, okay, and then again they will be re-esterified to triglyceride, phospholipid and cholesterol ester. These lipids along with the ApoB48, they are taken up by our lymphatic system, and through the thoracic duct, and ultimately they will come in the blood circulation. This is our blood circulation, which contains triglyceride, phospholipid, cholesterol ester and B48. This is called as a nascent chylomicron. So this role of chylomicron is to transport the dietary lipids, the lipids that we take in the diet, from intestine to the peripheral tissue, like adipose tissue, heart muscles, skeletal muscles. Right? So here in the blood, ApoB48 along with the triglyceride phospholipid cholesterol, they will form the nascent chylomicrons. Nascent means newly synthesized. Right. So if I draw the diagram again, so this is, suppose this is the blood circulation that contains the triglyceride, phospholipid, cholesterol, and B48. That is a nascent chylomicron. Then they will get the ApoC2 and ApoE apoprotein from HDL. HDL is a repository of apoproteins. So the ApoC2 and ApoE from HDL is given to the nascent chylomicrons, and then this phospholipid, triglyceride, and cholesterol ester along with the ApoB48, ApoC2 and ApoE, this is called as a mature chylomicrons. Okay. Now this mature chylomicron will go to the peripheral tissue. Which are the peripheral tissue? Like skeletal muscle, heart muscle, okay, adipose tissue, and in the capillary of this peripheral tissue there is a endothelium where heparin bound lipoprotein lipase enzyme is there, right? And once this mature chylomicron comes to the capillary, the ApoC2 of the mature chylomicron will activate lipoprotein lipase. This is frequently asked, which apoprotein will activate lipoprotein lipase enzymes. We have already learned about the lingual lipase, gastric lipase and pancreatic lipase in digestion and absorption of lipids. Here one more lipase is there, that is called lipoprotein lipase, which is activated by the ApoC2. Okay, this ApoC2 will break down the triglyceride this triglyceride will be broken down to glycerol and fatty acid. This fatty acid is taken up by the peripheral tissue like skeletal muscle and adipose tissue. They will skeletal muscle and heart, they will utilize it for the energy purpose, while adipose tissue will utilize fatty acid as a storage form of triglyceride, while glycerol will be taken up by the blood and go to the liver. So now this mature chylomicron, the triglyceride is reduced and the content which is remaining, triglyceride will be less in quantity, cholesterol ester and phospholipid and ApoB48. Okay. Once the function is over, ApoC2 again go back to ApoC2 lipoprotein will again go back to HDL. Okay. And the remaining is ApoB48, ApoE along with this less amount of triglyceride and cholesterol ester. Okay. So you can see here less amount of triglyceride, cholesterol ester, okay, along with the ApoB48 and ApoE. ApoC2 go back to the HDL. This molecule is called as a chylomicron remnant. It is called as a chylomicron remnant. Now we have seen that ApoC2 and ApoE, they are transported from the HDL. The function we know about ApoC2, that it activates lipoprotein lipase. So what should be the role of ApoE? Why it is donated to the chylomicron? Because this in the liver there are ApoE receptor. So with the ApoE receptor ApoE will bind with this ApoE receptor of the liver, and then this chylomicron remnant is endocytosed. Okay, and it is taken up by the liver. This is how chylomicron will help in transport of dietary lipids, because dietary lipids are hydrophobic, insoluble in water. So apoprotein chylomicron, that is the lipoprotein that will help in transport of the dietary lipids, okay, from the intestine to the peripheral tissue, and ultimately it is taken up by the liver cell. And as it will provide the fatty acids to the adipose tissue for the storage purpose and also for the energy purpose for the skeletal muscle and heart. Okay, so if I show you the diagram, so you can see here the dietary lipids contain the triglyceride, major portion, small amount of cholesterol ester. ApoB48 along with the triglyceride and cholesterol ester, they will form the nascent chylomicron. Then ApoC2 and ApoE is donated from HDL and it will form the mature chylomicron. This mature chylomicron will go to the peripheral tissue or extrahepatic tissue like adipose tissue, skeletal muscle, and their capillary endothelium will contain the lipoprotein lipase enzyme. This lipoprotein lipase will break down the triglyceride to the glycerol and fatty acid, which can be used for the energy purpose or storage purpose, while glycerol is go back to the liver. Once the function of ApoC2 is over, ApoC2 is go back to the HDL and ApoB48, ApoE, that is called as the, along with the cholesterol ester and less amount of triglyceride, it is called as the chylomicron remnant, which is taken up by the ApoE receptor in the liver and they will be internalized. So this is about the chylomicron metabolism. So if I revise in short, the chylomicron, major lipids present in chylomicron is triglyceride. Major apoprotein that is present in chylomicron is ApoB48. Role of ApoC2 is activation of lipoprotein lipid. And main, it is the exogenous lipid. It play a role in exogenous lipid transport. We will learn in hyperlipoproteinemia if there is an enzyme ApoC2 is deficient or lipoprotein lipase is deficient, it will lead to the increased chylomicron and that will lead to the increased triglyceride in our body and it is lead to the xanthoma. This is clinical application about the chylomicron metabolism. I am going to talk about endogenous lipid transport, that is VLDL, IDL and LDL metabolism. VLDL is very low density lipoprotein, IDL is intermediate density lipoprotein and LDL is low density lipoprotein. Exogenous lipid transport is mediated by the chylomicrons. So from the dietary lipids, they are transported to the peripheral tissue from intestine is via the chylomicrons, while endogenously synthesized lipids like triglycerides, phospholipid, cholesterol ester that are endogenously produced in the liver, they are transported to the peripheral tissue with the help of VLDL, IDL and LDL. So we will see in detail now. So suppose in the liver, this is liver, in the liver there is the hepatocytes. In the hepatocytes there are cells in the smooth endoplasmic reticulum. Lipids are endogenously synthesized. Which lipid? Like triglyceride, phospholipid, cholesterol, they are synthesized. Now these endogenously synthesized lipids have to be transported to the peripheral tissue. Like skeletal muscle, heart or adipose tissue. In adipose tissue mainly fatty acid will be stored, while skeletal muscle and in heart it is required for the energy purpose. So endogenously produce triglyceride. Okay, suppose in the liver, triglyceride, phospholipid, cholesterol ester. The synthesis and secretion of this VLDL is similar to the chylomicron. So these lipids are hydrophobic. So for transportation it requires apoprotein. For the transportation of this it requires the ApoB100. Okay, ApoB100 which is produced by the liver cells. Okay, so this ApoB100, triglyceride, phospholipid and cholesterol ester, it is called as the nascent VLDL. Now it gets ApoC2 and ApoE from HDL. Okay, so HDL will donate ApoE and ApoC2. So now this triglyceride, phospholipid, cholesterol ester along with the ApoB100, C2 and ApoE. This structure is called mature VLDL. Okay, so in the circulation from HDL, ApoC2 and ApoE is donated, and now this nascent VLDL is converted to mature VLDL. So you can see now this mature VLDL, it contains ApoB100, ApoC2 and ApoE. It will go to the peripheral tissue like skeletal muscle, heart or adipose tissue. And in the capillary of this peripheral tissue, there is an endothelium, and endothelium heparin bound lipoprotein lipase. Now this lipoprotein lipase is activated by ApoC2 and it will convert the triglyceride of the VLDL to glycerol and fatty acid. This fatty acid is utilized while glycerol will go to the liver. And this triglyceride is broken down to glycerol and fatty acid. Now, and once the function is over, once triglyceride is broken down to the smaller molecules, then this ApoC2 will go back to the HDL. So the remaining portion, so the smaller amount of triglyceride, phospholipid, cholesterol along with the ApoB100 and ApoE, now this structure is called as a IDL, intermediate density lipoprotein, which has a less triglyceride than the VLDL, and ApoC2 will not be there. Now this IDL, okay, it is B100 and ApoE. Now this IDL will go to the liver. In the liver, there is a ApoE receptor. So there is a two fat of the IDL. That one, it is taken up by the ApoE receptor on the liver and endocytose, and 50% of the IDL, 50% will endocytose and 50% are acted by the hepatic lipase enzyme. Hepatic lipase enzyme that is present in the sinusoidal surface of the liver. So what this hepatic lipase will do? It will convert the triglyceride to remaining triglyceride to glycerol and fatty acid, and only thing remaining will be cholesterol ester, some amount of phospholipid and ApoB100. And ApoE again will go back to the HDL. Okay, so this portion which contains mainly the cholesterol ester, no triglyceride and ApoB100, that is called as a LDL, low density lipoprotein. Okay, understood. Now this LDL has a two fat. Okay, this is LDL. Either it can go to the liver. In the liver, there is a LDL receptor. Okay, so this LDL will be taken up by the LDL receptor, and there will be endocytose, and which will be useful to provide cholesterol, and cholesterol will be converted to bile salts for the digestion of the lipids. Okay, so 70% of the LDL will go to the liver and it will be utilized, while 30% of the LDL, it will be taken up by the peripheral tissue. But which are the peripheral tissue that requires the cholesterol? So one is adrenal gland which requires cholesterol for the synthesis of steroid hormones. Okay. One is gonads. It requires for the formation of sex hormones. Then skin, that will take up the LDL. They have a LDL receptor and it will take up the LDL because here we require the synthesis of vitamin D. Okay, all the peripheral tissue that requires the cholesterol, there is a LDL receptor, and LDL will transport the cholesterol to the peripheral tissue, right? So we see the diagram also. So triglyceride, cholesterol ester and phospholipid, they will be incorporated with the help of ApoB100 in the liver, that is called the nascent LDL. In the circulation, ApoC2 and ApoE is donated by the HDL, and once ApoC2 and ApoE is along with the ApoB100, it is called as a mature VLDL. It will go to the peripheral tissue and capillaries, endothelial cell contain lipoprotein lipase. Okay, and ApoC2 will activate lipoprotein lipase and break down triglyceride to glycerol and fatty acid, and remaining portion is called as the IDL. This IDL has a two fat that 50% is taken up by the ApoE mediated endocytosis, while 50% with the help of hepatic lipase it will be converted. Triglyceride will be broken down to glycerol and fatty acid, and only cholesterol ester will be there, and ApoB100. So only ApoB100 present, if only apoprotein present in LDL is ApoB100. Okay, so this LDL has a two fat. One, it 70% is taken up by the LDL receptor present on the liver cell, while 30% will go to the extrahepatic tissue like adrenal gland, gonads or skin, where it is taken up. Okay, so you can see again here in the liver, the triglyceride and cholesterol, they will bind the ApoB100, it is called as VLDL, it gets ApoC and ApoE from HDL, it is called as mature VLDL. This mature VLDL will go to the extrahepatic tissue or peripheral tissue where it is activated by the lipoprotein lipase. It will break down triglyceride to glycerol and fatty acid. Once the action is over, ApoC2 will go back to the HDL, and remaining portion along with ApoB100 and ApoE, it's called the IDL. IDL, they have a two fat. Either it can go to the by the ApoE receptor mediated endocytosis, go inside the liver, or with the hepatic lipase enzyme, it is converted to LDL. LDL is a two fat. 70% will go to the LDL receptor present in the liver, while 30% will go to the extrahepatic tissue for the formation of steroid hormones and vitamin D. Okay. So if you can differentiate between exogenous and endogenous transport, so dietary lipids, they are incorporated into the chylomicrons, and at the peripheral tissue capillary, lipoprotein lipase will break down triglycerides to glycerol and fatty acid. It will be formed the chylomicron remnant, which is taken up by the ApoE mediated receptor endocytosis. While whatever endogenous reproduced lipids, they are with the ApoB100, they will form the VLDL, taken up by the capillaries of the peripheral tissue, and lipoprotein lipase will break down the triglyceride, and ultimately IDL is formed. Then IDL will be 50% is taken up by the liver, while 50% by the hepatic lipase it is converted to LDL. LDL has a two fat. 70% will take up by the LDL receptor present in the liver, and 30% is by the peripheral tissue. Okay. So in normal, this LDL that is produced is taken up by the LDL receptor. But if there is a excess amount of dietary cholesterol, then cholesterol repression of LDL receptor, cholesterol will repress the LDL receptor synthesis, and that will lead to accumulation of LDL. Or in some case, there is a genetic defective LDL receptor. In that case also LDL is increased. And if increased LDL, they are acted by the free radical, and it will form the oxidized LDL, which will be taken up by the macrophage, and they will form the form cell. And they form cell, ultimately they proliferate, and they will form the plaque, and they will block the lumen of the artery, and it will lead to the infarction. So this is all about the VLDL, IDL and LDL metabolism. High density lipoprotein metabolism. Okay. So here in the intestine as well as liver, so intestine and liver, both will produce the high density lipoprotein. So high density lipoprotein has various apoproteins like ApoA1, then we have learned in chylomicron and VLDL metabolism that ApoC2 and ApoE. Okay, and some amount of cholesterol ester is there. Okay, there are two important enzymes that are associated with this HDL, that is LCAT and cholesterol ester transport protein. Okay, that we will see. So by the liver and intestine, they will produce the HDL, which is discoidal in shape, and it is a nascent HDL. Right? Now, in the peripheral tissue, there is a SRB1 receptor, ABCA1 or ABCG1. They are ATP binding cassettes. Okay. These transport proteins are present in the peripheral tissue. What they will do? They will transport the cholesterol from peripheral tissue to the circulation. So all this protein, mainly the ABCA1, is responsible for efflux of cholesterol from peripheral tissue in the circulation. This cholesterol is taken up by the HDL, okay, and in the circulation, and there is a ApoA1, okay. This ApoA1 apoprotein will activate circulating LCAT enzyme. LCAT. Now, what is the full form of LCAT? It is lecithin cholesterol acyl transferase. So, as the name suggests, there is a transfer of acyl group, fatty acid. Okay, and lecithin, we know the other name of lecithin is phosphatidylcholine chloride. At the second carbon, there is a polyunsaturated fatty acid. So, it will take up the fatty acid. This LCAT enzyme will take up the polyunsaturated fatty acid from lecithin and it will bind with the cholesterol. Okay, and cholesterol with fatty acid, it is called as the cholesterol ester. So, with the help of ApoA1, LCAT is activated and it will convert the cholesterol inside the HDL to the cholesterol ester. Okay, so this is called HDL3. And with the more and more effect of this LCA enzyme, more and more cholesterol ester is produced, then it will be called as a HDL2. Okay, and this HDL3 and HDL2, what is the difference between these two? In HDL2, there will be more cholesterol ester as compared to HDL3, and now it turns to the spherical in shape. When there is too much amount of cholesterol ester, then it will inhibit the LCAT enzyme and further prevent the formation of cholesterol ester. Okay. So this is one, and ultimately this HDL2 that will be taken up by the SRB1 receptor. So SRB1 receptor is present at two sites, at the liver function. One at the liver for the endocytosis of HDL2 and another at the peripheral tissue. So at the peripheral tissue there is ABCA1, ABCG1 and SRB1 receptor. They will help in efflux of cholesterol from tissue to the circulation. In the circulation, LC with the help of ApoA1 and LCAT, cholesterol is converted to cholesterol ester, and when more and more cholesterol ester is formed, it is called as HDL2, which is taken up by the SRB1 receptor. And this cholesterol ester that is present inside the liver, now it will be helpful for the bile acid synthesis. And this HDL2 and HDL3, when there is more accumulation of cholesterol ester, so they will inhibit the LCAT. So this cholesterol ester, they are given to the VLDL, Chylomicron and IDL. So cholesterol ester is given to the VLDL, IDL and Chylomicron, and in return triglyceride is given to the HDL2. Then this is called as this is with the help of cholesterol ester transport protein, CETP. Okay, and this HDL2 is acted by the hepatic lipase and it is converted to the triglyceride is utilized and remaining portion is called as again it is converted to HDL3, which has a less cholesterol ester. Okay, so again you can see ABC1 transport protein that is mainly responsible, ABCA1, ABCG1 and SRB1, that will efflux the cholesterol from peripheral tissue to the circulation. In the circulation, LCAT enzyme will convert it to HDL3. This HDL3 again acted by the LCAT and it is converted to HDL2. Then hepatic SRB1 receptor mediated endocytosis occur, right? And this is all about HDL metabolism. So you can see that HDL will transport the cholesterol from peripheral tissue to the liver. That's why HDL called as a good cholesterol. Good cholesterol. While LDL, it will transport the cholesterol from liver to the peripheral tissue. Liver to the peripheral tissue. That's why it is called as a bad cholesterol. Okay. So whenever there is a defect, excessive cholesterol in the diet or defective LDL receptor, it will lead to increased cholesterol level. That will lead to increased LDL. That will be oxidized LDL, and it will form the plaque and form cell, and it will block the lumen. Okay. So this is all about the HDL metabolism. I hope you find this video helpful. Thank you for watching. Bye.